Hello students, in a molecular basis of inheritance, we were seeing about the HGP, otherwise Human Genome Project, we were discussing in the previous class. Human Genome Project is a mega project, I told you. It took around 13 years for getting completed. What is Human Genome Project means? It is the sequencing of nucleotide of the human genome. So sequencing the human genome, nucleotide by nucleotide, is called as Human Genome Project, we can see. Nearly 6 countries have participated in that work or project, and from 1990 to 2003 it was carried actually. In the previous lecture, I was telling you about the methodologies employed in Human Genome Project. Methodologies of HGP, I have to tell you here. In the previous class, I have told you some point related to this methodology also. What is the procedure, what is the step taken by the scientist for annotating or for sequencing the nucleotide of the human genome? Here you have to remember, genome refers to 22 autosomes plus 1 X chromosome and 1 Y chromosome, we can say. Totally 24 chromosomes have to be deciphered. Sequence of their nucleotide should be deciphered actually. It is a more important task, we can say. 3 into 10 to the power 9 number of base pairs are present in that genome. So much of genome must be, or so much of nucleotide must be sequenced properly. For this, 2 methods are employed by the scientist. One is called as EST, we can say. Another one is known as SAT, we can say here. EST refers to expressed sequence of or expressed sequence tags, they can be called. Expressed sequence tags are called EST. What is that expressed sequence means? We cannot decipher or we do not decipher all the DNA, all the nucleotides of the DNA here. We know one point. There are introns and exons present in the mRNA, same thing in DNA also. So that introns are not expressible, exons are the expressible sequence of the DNA. Here we are doing the expressible sequence only, we are analyzing actually. So in EST method, we focus upon exons mainly. Expressible sequence or expressed sequence only are focused. How they are focused, what is the method done here means? When exons are there, they will exhibit, they will produce the mRNA. Of course, with intron and exon, mRNA will be produced. That exon will be expressing itself as a protein molecule, remember. When we know the protein molecule, depending upon the protein molecule, we can say the amino acid sequence indirectly. When you know about the particular protein, what amino acid it has got, that we can decipher, we can easily find out. If you know the amino acid, easily you can find out their genetic code also here. We can find out the genetic code of that amino acid here. You know, genetic code dictionary. Which amino acid has got which code that we know. So based on that, in a reversible way, we can identify that genetic code. When genetic code is identified, easily you can identify the nucleotides of DNA also. This is the first mechanism where based on the protein you are going back to the nucleotides. You have already seen that one. DNA will produce RNA, mRNA. Genetic code is present in mRNA by a process called transcription. From genetic code only amino acid sequence will be determined. From amino acid only, protein molecules are synthesized. So, when you go in the reversible direction, you can understand the nucleotides of DNA clearly. So, by this method, you can sequence expressed sequences, you can sequence the nucleotides of expressible segments only. But that is not enough. When you want to go for human genome, you should go for complete introns also. For that, the second method is employed, that is called SAT method, it can be called. Sequence annotation technique, it can be called actually. Sequence annotation technique, that is the method employed for a second type or second method, we can say. So, to decipher the nucleotide in the DNA, we can go for sequence annotation technique. You must remember this point clearly, carefully, listen here. Remember, in this sequence annotation technique, the complete long chain of DNA molecule is taken actually. Now, the exon and introns we are focusing here, we take full long chain of polynucleotides of the DNA molecule. This DNA long chain will be broken into small pieces, otherwise fragments actually. How to break the DNA molecule into fragments? For that we need one enzyme called as REN, otherwise restriction endonuclease, we can say. When you get the fragments, remember this point, you are getting so many fragments. These fragments will be added, or these fragments will be linked to some vectors actually. The second step involved here is vectors will be joined with those DNA fragments. Otherwise, those DNA fragments will be linked with vectors. What are vectors? They are also nothing but small DNA molecules only, but they will be the carrier of this human genome, we can say. They will carry this human genome. For example, we can create artificial vectors like BAC and YAC like that. What is BAC? Bacterial artificial chromosome, yeast artificial chromosome. They can be synthesized in the laboratory condition. They are artificially synthesized. With those synthesized artificial vector or vehicle, our DNA fragment will be added, linked. For linking them, you need one enzyme. The name of that enzyme is called ligase. You may be remembering. REN acts as a molecular scissor. It will cut the DNA into fragments. Ligase acts as a molecular stitcher. It will join the DNA fragments. We have heard about the ligase already, you may recall. In a replication, I told you about Okazaki fragments. The newly synthesized strand away from the replication fork will be in fragments. How those fragments are joined later means by means of ligase enzyme only. So it is acting as a molecular stitcher. By using that stitcher, you will stitch the vector plus fragmented DNA here. Then this vector plus fragmented DNA will be allowed to multiply in a bacterial host actually. A host cell will be taken as a bacteria. A small or simplified prokaryotic cell will be taken. There we will introduce our small fragment. Now it will be allowed to express actually. Bacteria will express that protein or express that gene. By getting that protein, you can go once again go for the nucleotide sequence. This mechanism, you are allowing the bacteria to express itself. Whatever fragments are given to them, they will multiply and express. Now, after getting expression, you can match those DNAs or that expressed proteins in overlapping areas. Because you have made it into fragments, this fragment will overlap with this area, that overlapping sequences will be identified. For all these techniques, we need a computerized technique or computerized computational ideas also. One scientist called Frederick Sanger. Here, remember, Frederick Sanger. He is the scientist who discovered the method of deciphering the protein molecule like that. Amino acid sequence could be deciphered from the protein molecule. Same technique, Sanger method, was used for deciphering the nucleotide sequence also. So that is where we are applying some computer knowledge, otherwise data collection, everything. So such a 3 into 10 to the power 9 base pairs could be easily deciphered by means of this idea. Now this is called sequence annotation technique. Here you are not concentrating on expressed sequence. You are concentrating on all the sequences, complete gene, complete DNA you are taking. It is a polymerized chain. That chain will be broken into fragments. Then it will be added to a vector. Vector will be given to a bacterium. Bacterium will multiply that gene. Whether it is intron or exon, bacteria does not bother. It will start producing proteins or it will start to express. Based on that, you will come to understand the nucleotide sequence. This is called sequence annotation technique, it can be called. Frederick Sanger introduced this idea. Here you have to understand about BAC and YAC also. They are artificially synthesized chromosomes called bacterial artificial chromosome, yeast artificial chromosome, like that. So this is regarding methodology. In a 3-mark question it can be asked, what are the 2 methodologies used for deciphering the genetic code like that? One is called expressed sequence tag. Another one is called sequence annotation technique, it can be called. Make note of this, we will continue further about this genome project. Okay, students, let me continue with another subheading related to human genome project. Please listen to me. In the human genome project, we have seen what is the goals, what are the goals of that project we have seen. Goals of HGP we have seen here. Then I told you about methodology involved in HGP. What methods are used for identifying the human genome like that. Before that you must understand what is that genome means. 22 autosomes plus X and Y we can see. Now students, let us see what are the contributions made by the HGP. Otherwise, it is a mega project I told you. After working for 13 years, what are the discoveries made by HGP team or the scientists of HGP we can find out here. It can be called as salient features. In your textbook, it is given as features of HGP, it can be called. Salient features of human genome project we are going to study. So what discoveries were made, what contributions were given by the HGP or this project we are going to discuss. Based on that 13-year-long work, scientists have discovered that the human genome is made up of 3,164.7 million base pairs in it. There are 3,500 164.7 million base pairs present in the human genome. That is the first information given by this project. So, human genome contain how many nucleotides? 3164.7 millions of base pairs are present here. Okay. Second point here is the human genome is containing about 30,000 genes in it. About 30,000 genes are present in human genome, they discovered actually. Previously, before this project, scientists were believing that human genome must be having somewhere between 80,000 to 1 lakh genes in it, like that. 1 lakh to 80,000 genes must be present in the human genome. That was the belief before the project, before 1990. But in 2003, it was proved that human genome is made up of 30,000 genes only. Somewhere between 20,000 to 30,000 they have told here. Okay? 20,000 to 30,000 genes only are present in the human genome. It was covered. And then another point if you see means, in an average, a gene will be containing about 3,000 nucleotides in it. That also was proven here. And an average gene, 30,000 are there, average a gene will be containing around 3,000 nucleotides in it. That is another proof. Out of this, the chromosome number 1 is considered to be the largest chromosome we can say. Chromosome 1. That chromosome number 1 is containing 2,931 genes in it. It is the largest chromosome we can say. 2,961 genes are present there in the first chromosome. Y chromosome is the smallest chromosome in our body. So human genome contains Y chromosome. It is the smallest chromosome. It contains only 231 genes in it. So remember, all the chromosomes need not have same number of genes in it. There will be chromosome Y is the smallest chromosome. It is containing only 231 genes. Chromosome 1 is the largest chromosome. It contains 2,931 genes in it. Among the 30,000 genes or 20,000 to 30,000 genes, if you see means, there is a gene called muscular dystrophin gene is present. Muscular dystrophin gene. This is the biggest gene, we can say, largest gene present in our chromosomes, dystrophin gene. It contains around 2,000 or 2,400 something millions of nucleotides in it. So many millions of nucleotides are, I'm sorry, this number you have to check, I don't remember exactly. More than 2,000 nucleotides or 2,000 nucleotides are present in this muscular dystrophin gene. So muscular dystrophin gene is the largest gene present in our body. Please check that number in your textbook. Okay, check it. So what are the points we have got? Salient features of human genome. It is made up of around 3,164.7 million base pairs. The human genome contains around 30,000 genes in it, not 80,000 like that. And each gene may be average containing 3,000 nucleotides, we can see. Chromosome 1 is the largest chromosome. It is made up of 2,931 genes. Chromosome Y is the smallest chromosome. It contains 231 genes. Muscular dystrophin gene is the largest gene. I am not talking, this is largest chromosome, this is largest gene. In this gene, more than 2400 nucleotides are present, we can say here. So check this number only, very important, okay? Largest gene it can become. Now let me come to some other point related to Human Genome Project. Scientists have discovered that more than 50% of genes are having unknown functions, they told here. 50% of our genes do not perform any function, or their functions are not yet discovered, we can say. We do not know why some 50% of genes are present in our cells or in our genome. Their functions are unknown, it is true. Not only this one, only 2% of genes are involved in protein synthesis. That is also told by the scientist. So, 2% of genes only are involved in protein synthesis. Remaining 98% are not involved in protein synthesis. That means they may be acting like a regulator gene, they may be found in the introns. You know that lacapron and all we have studied. So they are helping for the gene, or they may be non-functional, or they may be simply present in our body. They may be due to antiqueness, we can say. That means from the ancestor we derived them, we inherited them. But it is not useful at present for this environmental condition it may be. So they are ancestral properties, remaining 98%. They are not directly expressed as protein. They may be controlling genes, they may be regulatory genes, they may produce some constitutive proteins that are not different. So only 2% are expressed as for protein synthesis. One more very interesting point here is 99.9% of our genome is similar in all human beings. Remember this point. 99.9% of genes are similar, genome is similar in all human beings. So in every individual, any human being you take, our genome will be 99.9% similar only. Whether you take an American or an Indian or a Chinese or an Negro, whomever you take, African like that, they have got a 99.9% similar genome only. So all these billions of nucleotides are similar, except what? 0.1% only will be the difference, we can say. Try to understand this point. This is the basic point for DNA fingerprinting we are going to see here. Only in 0.1% we differ from each other. There are so many human beings with so many morphological features and other things. Our differences are lying in just 0.1% only. In 99.9% we are all similar only. So our genome shares similar nucleotides, similar sequence only. Only in how much area it is differing? 0.1% only. That 0.1% of difference make uniqueness in each human being. Why we are unique from each other means that is due to this just 0.1%. I am using the word just, but it is not just. Imagine, in 3 into 10 to the power 9 is the complete genome. In 3 into 10 to the power 9 nucleotides, 0.1% means how much? It will be 3 into 10 to the power 6 actually. So even though it seems to be just 0.1%, it is 3 into 10 to the power 6 nucleotides. That means around 6 zeros you have to put 30 lakhs of gene, 30 lakhs of nucleotide will be differing. It may, in percentage wise, it may look very simple. In 99.9% we are similar, but only in 1% we are telling. That 1% is how much? 30 lakhs. Remember this point. in so many nucleotides we differ. That 13 lakhs nucleotide makes our uniqueness actually. That is the discovery made by Human Genome Project, and they have found that 99% is this. One more interesting point is our human genome contains many repetitive sequences in it. Our genome contains several repetitive sequences in it. In thousands of areas, there will be repeated sequences of nucleotide. Repeated means what? AT will be repeated for 1,000 times. Adenine, thymine, adenine, thymine, adenine, thymine, like that it may be repeated. In some areas, C and G may be repeated for 10,000 times like that. So repeated sequences forms the characteristic feature of human genome. We do not know yet why those repetitive sequences are present. What are their functions? We do not know. But our genome is made up of several repeated sequences. So presence of repetitive sequences is another character of Human Genome Project. Next one point I can tell you here. In 1.4 million locations, we exhibit difference in single nucleotide quantity. Single nucleotide differences will be exhibited at how many places? 1.4 million locations we can see. In our genome, 24 chromosomes, in 1.4 million locations, we differ with only one single nucleotide. As we have seen in sickle cell anemia, we will be differing in only one nucleotide. That is also coming under this 0.1% only. So only one nucleotide difference is found in 1.4 million locations of our genome. Those areas are called single nucleotide polymorphism, it can be called, or they will be called as SNPs also. SNP refers to what? That single nucleotide polymorphism. So how many areas we are showing only one nucleotide difference? In 1.4 million locations, we show the difference with reference to only one nucleotide. There may be 2 nucleotide difference, 10 nucleotide difference, that is different. But in so many large areas, we differ with only one nucleotide. So imagine why human beings are called the same species, Homo sapiens, even though morphologically we look different. Our height is different, our color is, skin color is differing, our shape of the face or nose, everything differs. If you compare different country people, we show different morphology. Even though so many morphological differences are there, we are all kept in same species because of this 99.9% genetic similarity. We are able to interbreed among themselves. An American can marry a Chinese also. There is a possibility of interbreeding. So that becomes the species capability of interbreeding nature. And 99.9% genome will be showing similarity. So caste, creed and other things, they are all evolved by human mind. This is what the genetical similarity, 99.9% we are all similar. So remember, these are the contributions made by Human Genome Project. This is the salient feature of HGP, an important 3-mark question it can be asked. Let me repeat it here once again. Listen. There are 3,164.7 million base pairs are present in the human genome. These million genomes or base pairs will make around 30,000 genes in our body or in our cells, we can say. Each gene will be made up of approximately 3,000 nucleotides. Some may be bigger gene, okay, that is different. Average, there will be 3,000 nucleotides in each gene, we can say. Chromosome 1 has got more genes. Around 2931 genes are present in chromosome 1. Chromosome Y has got only 231 genes. Muscular dystrophin gene is the largest gene. It contains more than 2400 nucleotides. Check this number, please. Then 50% of our genome has an unknown function. 2% of our genome only codes for protein. 99.9% of our genome is similar in all types of human beings. So we differ only in 0.1%. That is around 30 lakhs nucleotide we can see. Then I told you there are many repetitive areas in our genome. They have repeated sequences. In 1.4 million locations we show single nucleotide polymorphism. That means only difference in single nucleotide only. Instead of A, there may be there may be G-like. That is called the single nucleotide polymorphism. That is found in 1.4 million spaces or locations we can see. This is regarding the Human Genome Project. The discoveries made by Human Genome team we can say. And these are important 5-mark question. Sometimes 3 marks also it is mainly asked. You have to write at least 6 points for 3 marks. Be thorough about it. An important salient feature of Human Genome Project. We will continue about it after half a minute. Make it. Okay, students, after learning so many points about human genome, how we are going to use it, where we are going to apply this knowledge, that is very important. When you come to know about so many differences and everything, several applications can be made upon the human genome actually. Several ideas or scientific knowledge can be obtained from this data. This data can be computed and kept in a database also. Based on that only we came to know that 99.9% we are similar to each other. Now you see future applications of HGP. Future applications. That means where you can apply this HGP knowledge or Human Genome Project. Okay, this knowledge where and all you can apply, that we are going to see here. By understanding about human genome, we can understand the mutations happening in the human genome. We can find out. Mutations can be identified, whether any gene has undergone mutation or not, we can identify. Not only understanding mutation means you can understand the effects also. Effects of mutation can also be identified by this project. So which gene has undergone mutation, why a person shows that morphological difference, everything can be understood. That is how we have understood the sickle cell anemia and other genic disorders, phenylketonuria, all those things. When we come to another point, you can identify the behavior of cancer cells also. What is the modification of genome present in cancer cell? You know one interesting point, cancer cells have got ability to divide rapidly. Uncontrolled division of cells are happening in the cancer cell. So it will create a tumor, uncontrolled tumor it will create. So in tumor cells, how the genome behaves, what modification it has undergone, we can find out. When you understand that modification, genetic level modification, molecular level modification, you can try to prevent it by using some medicine or other things, so that cancer, origin of cancer can be prevented. That application is one more point, not only in tumor cells, even in other diseases, how our cells behave. The behavior of cells can be identified by this mechanism, behavior of cells. When you take a particular food material or nutrient, how our cells behave. Which nutrient make our body to produce certain hormones? Which nutrient will make us more energetic? All such events, all such ideas, knowledge can be identified or obtained by application of a human genome, we can say. Not only this one, the coordinated effect of coordinated functions of cells can also be identified by this project or by the knowledge of this project. If you want to perform, you remember in physiology, we have studied about digestion, respiration, excretion, everything. All these events or all these physiological functions are coordinated, interconnected with each other. If you can digest the food and get glucose, but without breathing, without respiration, you will not get oxygen. That oxygen will be used for oxidizing the glucose. So, respiration and digestion are interconnected. This kind of interconnecting behavior, it is called coordinated activity. Metabolism is a sum total of all biochemical reactions. How those all biochemical reactions are coordinated with each other, how they are orchestrated, we are told. How in an orchestra, there will be different musical instruments. Different people will play in a coordinated manner so that we can get a good song actually. In the same way, there will be orchestrated functioning of the cells in our body. That functioning you can understand, so that how a person behaves in a particular disease, how a person behaves in a particular age, everything you can identify, you can apply for future thinking. Then desirable modifications can be made in the human genome also. By applying this knowledge, you can make desirable modifications in genes. Remember this point. In our genome, we can make desirable modification. You can induce some mutation according to our wish. So in future, if a couple want only tall children means, that can be done by manipulating the genome possibly. Only a couple want a particular type of nose for the children means, that can be done here. So by altering the gene, you can make mutation, you can get desirable modification, desirable changes in the morphology. These are called applications of HGP, applications of Human Genome Project, it can be called. So remember students, these are the Human Genome Project, their application, their salient features and methodologies, goals of HGP, what is HGP, 5 points we have seen. Definition for Human Genome Project, goals of Human Genome Project, methodology involved in that Human Genome Project, salient features of Human Genome Project, and the future application of that project, everything we dealt in detail. Now I am going to explain about one of the applications here. One of the applications of Human Genome Project is DNA fingerprinting technique, it is called. Let me give you some introduction related to DNA fingerprinting. Later we shall continue it in the other lecture. Just to make note this for 2 seconds, I will come back. Okay, students. Next to Human Genome Project, we must have some idea related to DNA fingerprinting. We have to see here. DNA fingerprinting technique we have to see. Just from the name itself you can understand. You know, we are differing from each other, we exhibit variation. Even the children born to the same parents will have differences. Morphologically, those differences could be identified by our thumb impression, you may be knowing. Our thumb impression will be differing from person to person. That is called fingerprinting, normal fingerprinting. Same kind of uniqueness is found in the DNA also. That identifying the uniqueness with reference to DNA is called DNA fingerprinting technology. So what is the DNA fingerprinting? It is a method used for identification of uniqueness of an individual at molecular level, we can say here, at molecular level. So in DNA level or at molecular level, how to identify the uniqueness of an individual. That is called DNA fingerprinting. It is otherwise known as DNA profiling also. It is also known as DNA profiling. That how to identify this profiling or fingerprinting we have to see. Already I told you, in 99.9% we are all similar in our genome. Difference will be in how much percentage? 0.1% only will exhibit the uniqueness in our body. 99.9% we are similar with each other. Only in 0.1% we show the difference or we show our uniqueness, we can see. How to identify that? Our aim is to understand the difference. In DNA fingerprinting, what is our aim means? How we are differing, in which point we are differing. Why our fingerprint differs? That is not a fingerprint, this morphological thumb finger here. It is about DNA, molecular level fingerprinting it is called. How it is differing, how that uniqueness is caused, we have to see. You see, already I told you, even though it is 0.1%, it is not a negligible number. It includes 3 into 10 to the power 6 number of nucleotides in 3 into 10 to the power 6. That means 3, 6 number we have to take. That indicates 30 lakhs of nucleotide differences. Identifying this 30 lakh of nucleotide is called DNA fingerprinting. So it becomes a very cumbersome project or very complicated project. How to identify the 30 lakhs genes or 30 lakhs nucleotide. That becomes the process of DNA fingerprinting. About that, we will see in the next class. Today just I gave you some introduction. You could have understood the idea of DNA fingerprinting. It is not searching for 3 into 10 to the power 9 nucleotide. It is very difficult to take so many nucleotides, sequencing them, comparing them, everything. You have to compare only 1.1%. Even if it sounds 0.1% negligible, it includes 30 lakhs nucleotide. So again 30 lakhs nucleotide also becomes a large measure. For that we use another technique. That is called DNA fingerprinting, which we will discuss in the later class. Let me stop with this today. Today I told you about methodology of HGP, then salient feature, then future application of HGP. I gave you introduction about DNA fingerprinting. Tomorrow or in the next class, this lesson will be completed. Okay, this molecular basis will be completed. Let me stop here. Thank you.